So the next unit that we are going to move on to is chapter 12 in the textbook, and it's covering DNA, DNA transformation, and the central dogma. So if you kind of remember the central dogma from pre-AP bio, then this might be a little easier for you to wrap your head around since it's a lot of review information with just a little bit of detail added. So first off, chapter 12, Summary and Learn Smart will be due next Friday. And we're mainly going to introduce the chapter today. So there's not too much stuff. There's a little bit of notes and there's a little bit of an assignment for you to just kind of summarize what you understand from today. So there's vocab that I need you to get down in your notes. And the first word is pathogenic, which is causing or capable of causing disease. The second one is harmless, obviously the opposite of pathogenic in this case. It would be about not being able to or unlikely to cause harm or damage to another person or a thing. A plasmid is a small extra chromosomal, which means it's not part of the original DNA set. It's an extra chromosomal DNA molecule within a cell that is separate from the chromosomal DNA and can replicate independently. It's most commonly found as a small double-stranded circle of DNA in bacteria. Most other organisms don't have it. It's most commonly found in bacteria, and it's super useful to have. Transformation or bacterial transformation is the genetic alteration of a cell of bacteria by taking in genetic information from the environment through the cell membrane and incorporating into its own genes. So this is basically saying that you can take any DNA that you find in the environment and incorporate it into your own DNA. However, we can't do that as people, but bacteria actually can do it. And that's how a lot of times they get new genes or new combinations of genes because they don't do things like recombination and crossing over. Dnase stands for deoxyribonuclease. There's an ACE ending and ASE ending, so it's an enzyme, and it catalyzes the hydrolytic cleavage of phosphodiester linkages in the DNA. Basically, it breaks down the backbone and it breaks apart DNA. Rnase, on the other hand, is very similar, except obviously it applies to RNA, right? So its name is ribonuclease. It's an enzyme that catalyzes the degradation of RNA into smaller components. So when you break RNA apart from the backbone, the sugar phosphate backbone, it's breaking into smaller pieces. You're not necessarily damaging the nucleotides, but you're making sure that they're not attached to each other anymore. And the DNase works the same way, but they are different because DNase only works on DNA and RNase only works on RNA. And the likely reason is just because the structure of a double-stranded helix versus a single-stranded RNA is very different, so they have different enzymes. Protease, notice again, it still has an ACE ending. It's an enzyme that breaks down proteins and peptides. And lastly, transgenic organisms are any genetically modified organism whose genetic material has been altered using genetic engineering techniques. And that could be from bacterial transformation, it could be PCR, it could be, we know CRISPR that we have that we can actually alter the human genome with. We don't actually do it, but we could. So that's terms that you want to know going into this chapter. Your homework, other than the chapter 12 summary and learn smart, that I highly suggest you doing early rather than doing at the last minute, is that you're going to be writing a one-page summary explaining 2 main experiments that helped us discover that DNA was the genetic material of life and how DNA is the transformation factor that bacteria will be using and intaking during the process of when they do their version of genetic recombination. And our learning objective is that you all will be able to justify the claim that humans can manipulate heritable information by identifying at least 2 commonly used technologies. And so we're going to go through basically one of them. And the background of the experiments is using a certain bacteria that is infectious and a certain bacteria that's not as strongly infectious. And we usually test on mice in labs because they are easy to breed, they're cheap to raise, and they're a little bit less humane of an animal. So humans have this weird idea that if we need to do live testing, we don't want to test on humans because we don't think it's ethical. And a lot of people also agree that it's not ethical to test on other animals either, but that's usually on animals that have higher intelligence like chimpanzees and dogs and pigs. So a lot of times the first phases of live testing animals will be on rats or on mice, even though they are quite intelligent animals. It's just because of the ease of access and because they reproduce really fast and they're just slightly further away from humane than from like humanity and intelligence levels of humans. So in the first basic experiments, they injected a group of mice with strain S, which is just a strain. It's just a type, a family. So a group of bacteria that is known as strain S, and this actually causes the mice to die because the highly infectious form of the bacteria, because it has a capsule on the outside. And the capsule protects it from the antibodies and the white blood cells in the mice's body. And then on the other hand, they also injected another group of mice with strain R. And this bacteria doesn't have the capsule, so it doesn't have this protective layer that protects it against the animal's immune system. And so the immune system can fight back, and the mice actually stay alive when they're injected with strain R. They also tried and injected heat-killed S strain bacteria into mice. So basically, they cooked the S strain bacteria until it was no longer alive and injected into mice, and they saw, did the mice get sick or not? And turns out the mice don't get sick from this and they don't die from it. Okay, and then the last one that they tried that was related is they injected heat-killed S strain. So they took the S strain that they already killed by heating it or by boiling it, and they also put in live R strain, right? We know that R strain that's alive doesn't kill mice, and we know that S strain that's dead also doesn't kill mice. Okay, but for some reason, when you inject them together, the mice actually still die. And so when they drew the blood from the mice, they actually found live S strain bacteria, even though there was no live S strain bacteria initially injected into the mice. And so this was just kind of interesting to be like, well, where did this S strain bacteria come from? And why did the mice die in the first place? Because they were injected with two things that don't actually kill them and are proven to not kill them by other experiments. And so this lab provides an evidence that DNA can transform bacteria. So the discovery of the genetic role of DNA began with research by Frederick Griffith in 1928, and he worked with two strains of bacterium. One was pathogenic and one was harmless. And so when you go back here, the S strain is pathogenic because it is highly infectious and kills the mice, and the R strain turns out to be harmless because even though it is infectious, it isn't able to kill because it doesn't have that protective capsule that prevents it from getting eradicated by an immune system of an animal. So the initial experiment, as you can see here, I don't know why I have these boxes here, is that when you have the encapsulated S strain, the results are that the mice actually die when you inject them. When you have the non-encapsulated R strain injected into mice, the mice actually survive. Now, if you boil the S strain until it dies and you inject it into the mice, it doesn't kill them either. But if you put in the boiled S strain with the R strain, neither of which by themselves have the ability to kill mice, the mice actually still die. And when you extract a bacterium from the mice's body, you actually find live S strain. Super, super strange. Okay. Yeah, this is basically just going over. So this is kind of interesting to think about because where is this S strain bacteria coming from? And if we were to take the results of these and we incubate the bacteria with Rnase, Dnase, or Protease, what would we find? The idea is basically saying, if we put Rnase in here, in the process of the bacteria turning into S strain bacteria, anything that has RNA is going to get broken down by the enzyme. So then no RNA is going to be present. So transcription can't happen. And then the central dogma process can't happen. Okay. That means that no proteins are going to be made in the end. On the other hand, if you put Dnase in, it means that any DNA is going to get broken down. So if DNA is broken down, you can't even start the central dogma and you can't even move DNA because it's all getting broken down. If you put protease in, you can go through the central dogma and you can go from DNA, transcription to mRNA, and then translation. But the proteins themselves, after they're made, they're getting broken down by the protein enzyme, which is protease. Okay. And so there is an experiment, a very, very famous experiment that was performed. And you guys are actually going to write a short essay on it. And so in the post below this one, after you turn in these notes that you just took on the general experiment of the Griffith experiment, you're going to read about the overall discoveries from the site. I've linked it in the post again, like I said. You're going to watch the short explanation video of the Griffith experiment and the short explanation of the Avery, McCarthy, and McLeod lab as well. And like I just explained in the Griffith experiment, they show that something is moving, but there's no exact proof of what it is yet. And then the second experiment from Avery, McCarthy, and McLeod actually does explain how they decided what was the transformation factor and what was moving from the dead S strain into the live R strain to make the whole R strain turn into a live S strain. And so you're going to basically write a summary of what happens, and you're going to try to use the following vocabulary, which is DNA, smooth, rough, pathogenic, RNA, Dnase, Rnase, Protease, heat-killed, capsule, and harmless in your explanation of what happened overall in these experiments and why it contributed so much to science and what we know now about how DNA is the genetic material of life, and it's the transformation factor in bacterial transformation.